Genetic engineering. This is really where the fun begins. This is where you start to see, now, you understand the basics of molecular biology, you understand replication, transcription, translation, you understand a little bit about gel electrophoresis. Now, genetic engineering is really such a cool field of modern biology where you can now start to do some crazy things. What do I mean by that? I mean simply taking a foreign piece of DNA, let's just say from a human DNA, and inserting that into a bacterial plasmid. And this plasmid will be transformed into the bacterial chromosomes of the respective E. coli cells, and you can generate this really cool-looking plasmid, with your foreign piece of DNA, and your bacteria can now replicate this. Your bacteria now have the genetic instructions to code for whatever this codes for. This is really cool. Let's use insulin because it's always used, and it's a good example to use because we see some real applications here, or rather straightforward application. Let's say this section of the human DNA, this part codes for the human gene insulin. Insulin is a hormone that will lower the blood sugar levels and by transporting the glucose in the blood to the cells. And of course, this is prominent because we need these for diabetics. Diabetics, which we'll go into more detail on other videos when we do endocrinology. Diabetics need insulin shots. And we have to purify these insulin and provide. How many diabetics are there out in the world? There's a lot of people with diabetes. So how do we manufacture all these insulin for these diabetics? Very cool. The idea here is to take this foreign piece of DNA that contains the insulin gene instruction and insert it into a bacterial plasmid. Why do I want to do that? Well, the plasmids are self-replicating. For starters, plasmids are self-replicating. And bacteria in general have an exponential division. They do binary fission and they grow like this. I mean, they really grow fast. And your bacterial plasmids also self-replicate all the time. And the idea was, hey, you know what? Is it possible to put the human insulin gene instruction and put it in the plasmid of bacteria, and somehow can bacteria synthesize, maybe it doesn't have to be a pure form of insulin, but can it synthesize insulin? And the answer is yes. So, this is really cool, and they have done a crazy amount of things. They've inserted, many different genes into plasmids, and now we have all these, things that are used for cancer therapy and genetic engineering for fishes and food and agriculture. I mean, this field is really fascinating. But you'll understand and appreciate it at the end when we actually understand how to do this. Idea from far away, it looks simple. Hey, what we're going to do is take a foreign piece of DNA from the human gene, and we're going to insert it into the bacterial plasmid. Easier said than done. So let's try to go ahead and try to get a better foundation of understanding how is this carried out. And if you watch the gel electrophoresis video, you'll have a good idea. And this shouldn't be, a lot of the things I'll introduce should not be new ideas. We need restriction enzymes. We need restriction enzymes to carry this out. So here's my foreign DNA, or here's my gene of interest, let's say. Here's my gene of interest that I want to take out. So I really want to basically just take this piece of coding segment right here, this instruction right here, and somehow put it into my plasmid. Now, you know what? Over the years, not pharmaceutical companies, but biotechnical companies that sell plasmids and bacteria and enzymes and all these stuff have really engineered a plasmid, E. coli plasmids, that have a great advantage to making our life easier to inserting this into here. And what they do is they have a selectable target right here, noted as the AMP-R gene. So AMP stands for ampicillin, and ampicillin is an antibiotic. What are antibiotics? Antibiotics kill bacteria. Antibiotics kill bacteria. This is antibiotic resistant, ampicillin resistant. Why do we need this? Well, we'll talk about that just later on. Let's just understand what it has here. And there's many other things here, promoters and things that I'm not going to talk about because this is not, a high-level course. But this is simply, at an undergrad level, somewhat, you should understand just the basics. So we have an AMP-R region over here, and we also have a gene called the LAC-Z gene. And if you remember the LAC-Z gene from when we did prokaryotic gene expression analysis, you'll remember that the LAC-Z gene synthesizes an enzyme called beta-galactosidase. And beta-galactosidase can take the lactose and hydrolyze it and break it apart. So this guy right here codes for beta-galactosidase. This functional plasmid right now codes for beta-galactosidase, and it can break down lactose. And we've engineered in such a way, not we, but the companies have engineered this region right here in such a way that we're able to selectively target a lot of restriction enzymes. Different sites of the LAC-Z gene, or over here even, doesn't matter. Let's just say this region over here makes an EcoR1 cut. This region right over here. Once again, remember EcoR1, it's a restriction enzyme. It will make a staggered cut. That's just over here. And over here we can have different enzyme. Let's say BAM, H1, HIND3, maybe a hint over here. But let's not make things complex, let's keep it simple. Let's use EcoR1. You like EcoR1. The EcoR1 will be the respective enzyme that will be used to make a cut and disrupt the LAC-Z gene. Keep that in mind. We will also use EcoR1 in our gene of interest. Why do we want to use the same restriction enzyme? Well, if you remember, EcoR1 recognizes GAATC and creates a staggered cut. This part right here, when they're separated, these parts are referred to as the sticky ends. These parts are referred to as the sticky ends. Why? They can, of course, bind back together, right? They can religate back together very easily because they match up. They're kind of like glues now. So if we make a single cut over here, what's going to happen? We're going to make this plasmid linearized now. It's going to kind of open up now, right? So let's just say it opens up something like this. And we have our AMP-R over here. And we have a part of our LAC-Z gene over here. And a part of our LAC-Z gene over here. And we have the cut over here. Our sticky ends over here are still there. They can actually religate back together. They can religate back together. So hold on. So we have sticky ends. We've made a restriction cut by introducing a restriction enzyme called EcoR1. We've also, in another microcentrifuge tube, in a separate tube, we have our gene of interest. We introduce the enzyme EcoR1 to the restriction enzyme. We have a nice cut. And now what happens? This fragment right here should fit in very, very nicely to this part right here because they have compatible ends. Very important. They have compatible ends. So this is called a two-piece ligation. This, by the way, is called our vector. This right here is our vector. This is called our insert. We want to successfully insert our gene of interest into our vector and make a nice working replicative plasmid. So okay, let's just say this happened, okay? Let's just say, okay, so we have our nice gene of interest now. Very, very cool. We add ligase. Very nice. Very cool. Great. Now, I want to introduce something just a little bit. Let me introduce something before we move on. Remember when we just talked about when the EcoR1 makes this cut over here? When EcoR1 cuts over here and this plasmid is now linearized, we have a pretty good chance of having it relinear. We have a chance of it coming back together by religating. Once again, they're sticky ends. They're compatible with each other. They can go back with each other anytime they want to. To prevent this from happening, we use something called the sipping method to sip the ends of the sticky ends. That means we remove a phosphate group. Remove a phosphate. And by removing the phosphate, now we allow this fragment, this nice one, open fragment, to not come back together. It's very, very important. But now when we have this and we added ligase, they'll just phosphorylate the ends, and now we'll have a nice working plasmid. So I'm just trying to give you some of the insights into the problems that we face. It may seem easy when I'm explaining this to you, but I mean, trust me, it took many, many years of modifications of these biotechnology tools and genetic engineering and how to make it really effective. So we use this method on the vector, not the insert, the vector, to make sure it does not come back together. The gene of interest will hopefully insert itself, and now we should have one nice fragment. And you might say, hey man, okay, well how do you know? How do you know that the gene of interest has gotten in here? How do you know? Very interesting. Well, there's several ways of knowing. The first way of knowing, really, is to run a gel electrophoresis test. You want to run a gel chamber, you want to run a gel box, and you want to introduce, so once you have your plasmid, usually what you want to do if you're smart is you want to just take your insert, measure how big it is. You want to take your plasmid, measure how big it is. You want to take your plasmid with the insert vector, or you want to take your vector with the insert, and you should see something like this. Your insert should be smaller, your plasmid should be slightly bigger, but your plasmid and the insert should be a bigger fragment size. So you can kind of see, hey, do I have the appropriate things by running a gel box? By running a gel test, I should be able to see that, hey, look, if I introduce an enzyme into this box right here, I should see now two fragments. I should see the insert come out, and the insert should be over here, and the plasmid should be somewhere over here. So the gel test really gives you an idea of, hey, whether or not this was integrated properly. Do we have the right base size, and so on and so on. And you might say, okay, you know what, fair enough, okay, running a gel test, cool. What else? Why do we have this AMPR? Why do we care? Why do we disrupt this LAC-Z gene? Well, we're doing everything for a purpose. If we have successful integration of the LAC-Z gene, within the LAC-Z gene, if we have successful integration of the gene of interest into our LAC-Z gene, that means the LAC-Z gene becomes non-functional. LAC-Z gene is non-functional now. Meaning what? What does that mean? That means no beta-galactosidase. Beta-galactosidase. Meaning you will not hydrolyze lactose. That means that you have some kind of integration of insert in between the LAC-Z gene. This is good, you want this! You want this! And there's actually a test done. There's a test done by introducing a molecule called XGAL. And XGAL, if it's hydrolyzed by beta-galactosidase, if XGAL, this molecule, when it's introduced, if it's hydrolyzed by beta-galactosidase, the colonies turn blue. However, if the XGAL does not encounter beta-galactosidase, the colonies are white. What do you think you want? You want the white colonies. This is a very good test to run. Your white colonies should have the appropriate integration of your insert into the vector. The blue one, if you have blue colonies, that means that the LAC-Z gene is still functional. This is one way to compare whether or not you have successful integration of the gene of interest into your plasmid. So it's really, really cool. I mean, you can't just, look at it under a microscope and say, hey, I've got it, let's move on. This is now, the molecular level, so it's really, really cool. What else? And let's talk about the AMPR, the ampicillin resistance. I told you that we're gonna, well, you might not know this, but we have petri dishes and we have agar all over it, and agar serves as the nutrients. And we're gonna have these bacteria growing. These bacteria are growing exponentially, making colonies and colonies. And these guys, when they're introduced to a plate with ampicillin drug, any bacteria that does not have this selectable marker, the AMPR, will die out. Bacteria is dead automatically. However, if it's a functional plasmid, if everything works appropriately, it should also be transcribing the gene for ampicillin resistance. You now have these colonies that can withstand the ampicillin, and this will grow in an AMR positive plate. So here's your petri dish plate. I can't even draw, but here's your, it's a very bad drawing, but here's your plate. This is AMR, this is drug positive, and you should see colonies start to grow. If you run a test, the colonies should be white if you run the XGAL test, and this really, really tells you, hey man, you've successfully, most likely successfully integrated the insert into your vector. And now what do you have? You have a nice plasmid. You can reintroduce them into the bacteria, have bacterial transformation, and now the bacteria will divide. And transformation is really, really important. Transformation, remember, we did this at the beginning of molecular biology, or molecular genetics, I believe, yes. Transformation is when bacterial cells, or something will take up a foreign DNA. When we talked about it, we talked about the Frederick Griffith experiment and how we did transformation. In this context, we're going to say bacterial cells taking up foreign DNA. In this case, it's going to be human DNA. Bacterial cells take up foreign DNA, but this transformation rate is very low. The rate is very low, meaning that if you start, if you have conjugation of bacteria, it doesn't mean that all of them are going to have this successful plasmid that you've created. So this transformation is low, so now you really want to select for what can grow and what cannot grow. The bacteria that come up to be, that don't undergo transformation, well, they're dead, because they can't live in the AMPR positive plate. However, the bacteria that do have the successful plasmid for the AMPR resistance genes, these guys can start growing on this plate. So it's very, 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 very cool. This is basically how this plasmid works. This is the introduction to genetic engineering. Unfortunately, AP Bio does not have an extensive course overview of genetic engineering, but this is an insight into the field of genetic engineering. I really hope you've enjoyed this lecture, because this is an interesting lecture, and this gives you an insight into what can possibly be done. Go ahead and go on YouTube and watch genetic engineering and how they modify salmon, fishes, and all these things. Sometimes it's a bit scary, but it's very cool. So this is it for this video. Thanks for watching.